morning, everyone. Today, we will have a special lecture for the speaker. The speaker is Professor Ana Maria Otero Casal from Santiago de Compostela. This is the second time to talk about this, but very, very interesting for us. So she will talk about how to speak bacteria each other. Can you start this segment?
Thank you so much. I would like to, first of all, I would like to thank Sensei and the colleague in the HiSEP program for inviting me again to talk to you. Now? Okay. So, I would say that I would like to thank the埼玉University for inviting me to this HiSEP program and to talk about my passion, which is bacterial communication. But before talking about to you about how bacteria communicate to each other, I would like to introduce myself. I come from Santiago de Compostela, which is a small city on the north of Spain. So this is Spain, and Madrid is right in the center of the country. And Santiago de Compostela is right on the corner of the country, 500 kilometers far from Madrid. So if you see Europe, we are in the very far west of Europe. Romans used to call our region the end of the world, because at that time they thought that there was nothing beyond my region, which is called Galicia. And Santiago de Compostela is important and is well known in Europe because of the cathedral. This cathedral was built in the Middle Age, and many people in Europe at that time traveled, walking all through Europe to visit this cathedral, which is a holy place for Catholics. The city is not so big, but the university is quite big. It's one of the oldest universities in Spain and Europe. It is 500 years old. We have 23 faculties and a little bit more than 27,000 students. So the atmosphere in the city, as you can imagine, is very nice. Good place to study. And we are specialists in life sciences. So our medicine, pharmacy, and biology faculties are quite famous in the country. And you may wonder why I am in Japan. I came to Japan almost 20 years ago. And at that time I was interested in lipid synthesis by microorganisms. And the best laboratory in the world was here in Japan. So that is why I came. This is Murata Sensei laboratory. And you can see here, this is me, and this is Nishiyama Sensei many, many years ago. He hasn't changed so much. So, bacterial communication. Last year I was here, and I was explaining about how bacteria communicate using chemical signals, using molecules. But then one of the students, which was a physics student, asked me about physical communication. Do bacteria use wavelengths, electrical signals, or any physical signal? And the answer was yes, they do. So for this year, I have prepared a mixed lecture. So I'm going to talk a little bit about chemical communication again, which is the best characterized communication, but at the same time I'm going to speak a little bit about physical communication. This is new for me because I'm not a physical physicist, and I'm not a specialist on these physical experiments, so I hope you don't make too difficult questions at the end on physical communication, okay? So, bacterial communication. You may think that if I told you bacteria communicate, you may think, well, I'm not surprised, maybe normal, but 40 years ago, that was not expected. No microbiologists, no biologists could think that bacteria could communicate. At that time, even a Nobel Prize, very important Nobel Prize, Francois Jacob, thought that bacteria were very boring. They just live by growing and dividing, growing and dividing, growing and dividing, and that's all. Okay? But the news came from people that were studying very strange, very funny bacteria. Those are marine bacteria that produce light. Here you can see a plate in the dark, and they're producing blue light. So in the 70s, scientists became very interested in these bacteria, very fancy bacteria. And most of these bacteria live inside marine organisms, like squids or fish. They make their living with a mechanism called symbiosis. So the squid gives energy to the bacteria, and the bacteria produce light for the squid, so that they can hunt or they can search for cover. So we're going to see a very short video on how these bacteria produce light for the animals. In this case, a fish. And the fish has under their eyes a bag with bacteria. So if you turn off the light, this is what you see. The bacteria are producing the light for the fish. So, what was curious about these bacteria was that, first of all, how do they do? There's an enzyme called luciferase. We're not going to go into detail of the enzyme. It's a very complex enzyme that takes that oxidizes simultaneously two substrates with oxygen and produces light. Very particular mechanism that has been studied for years, especially in the 70s. Okay, but what was very special of these bacteria is that they did not produce light all the time. So if you see a curve on growing and light production, this is how bacteria grow continuously. Okay? But light is not produced continuously. So no light, and all of a sudden, all the bacteria decide to start producing light at the same time. So how do they do? How do they manage to communicate and decide we are going to produce light all together? So at that time, it was in the early 70s, people worked a lot on that, and finally, in the early 80s, they discovered that these bacteria were producing a small molecule that was released to the culture media. This small molecule is called acetylmorphin lactone, AHL, and goes through the cell membrane to the culture medium, and allows the bacteria to know how many bacteria are in the surroundings. So this is the model. One bacteria continuously produces a signal. This is the bacteria, and these are the signals. If very few bacteria, very few signals. And the bacteria knows she's alone, or very few bacteria in the surroundings, so it's not worth making light. But as bacteria continue growing, the number of signals accumulate in the media. And now the bacteria know that there is a lot of bacteria in the surroundings, probably inside the organ of the squid or the fish. So they decide to start producing light, expressing the luciferase and producing light. This was a very peculiar mechanism at that time, because that means that bacteria were behaving as a coordinated population. And nobody could think that that was possible for bacteria at that time. They thought, okay, that's a very particular thing for these special marine bacteria that produce light. And they need to coordinate because this consumes a lot of energy. But probably they are the only bacteria that can do such a thing. Okay? Still, these crazy marine biologists continue working on that. And they could identify the genes that were responsible for these. This is the luciferase operon, the chain of genes that codified the luciferase, and were among the first genes that were cloned when molecular techniques were available early in the 80s. So when they cloned the luciferase genes, they found that together, in the same operon, there were the two proteins that were responsible for this coordination. These two proteins are called LuxR and LuxI. LuxI is the receptor of the signal, and LuxI is the enzyme that produces. So now they have the molecular model for this. So at low cell density, the receptor is expressed, and the enzyme that produces the signal is also expressed. The signal goes through the membrane and is diluted away from the cell. Okay? But luciferase is not expressed. When the cell density increases, the signals start accumulating in the culture medium or inside the organ of the squid. And then the signal can bind the receptor. So the conformation of the receptor changes and binds to the promoter of the luciferase genes, and finally light is produced. So this was the model that was established early in the 80s. And again, nobody paid attention to that. They thought it was a very particular mechanism that only that bacteria could use. And it took 10 years for the scientific community to realize that maybe this mechanism is more common than what we think. So 10 years later, and simultaneously, in 3 different laboratories, they discovered that the same mechanism was being used to control 3 very important characteristics in 3 different bacteria. So antibiotic production in Erwinia, conjugation in Agrobacterium, and virulence in Pseudomonas, all 3 processes are controlled by AHLs. And at that time, they changed the name of the phenomenon. Initially, it was called autoinduction, and now they changed to quorum sensing. Why quorum sensing? Because all these bacteria, they were starting to do these actions only where enough number of bacteria were together. That is the meaning of quorum in Latin, minimum number. So this phenomenon was called quorum sensing, detection of minimum number. All right? And these were the signals, a family of signals called AHLs. All of they're small molecules that can go, most of them can go through the membrane directly without transportation, lactone ring and a tail. And the tail can change in the length. And every particular bacteria use only 1 or 2 signals that allow to communicate with the same species. All right? So now we know that more than 50 very important bacteria have the same mechanism, as you can see here. Virulence, motility, exopolysaccharides, antibiotics, motility. All those characteristics are controlled using the quorum sensing mechanisms. And even though we have some bacteria like E. coli, Escherichia coli does not produce a signal, but has a receptor for the signal. That means that E. coli is eavesdropping the conversations from all the bacteria in the environment and controlling very important traits like acid resistance or motility or biofilm formation. Okay? Just to give you an idea of how this works, Pseudomonas aeruginosa is a very important pathogen, human pathogen. And Pseudomonas aeruginosa, that has a very, very complex quorum sensing signals that includes AHLs, but also other signals, grows by producing biofilms. They attach to surfaces and then produce a thin biofilm where bacteria make their living and are protected from antibiotics. So this is a normal wild-type Pseudomonas that is growing attached to a surface. It's a pink surface. So you see a lot of growth. But if the quorum sensing genes are mutated, knocked down, then pseudomonas cannot form biofilm and therefore cannot colonize your lungs or your hearts, right? And another thing that we know now is that AHLs is only one of the languages that bacteria use. There are other molecules that can be used to communicate. So AHLs are used by a group of bacteria that are called gram-negative bacteria. Gram-positive bacteria, which are a large, different group of bacteria, use peptides, small peptides, that are also released to the media. And there is a very special signal called AI2. This small signal is a general, common language for many, many bacteria, both gram-positive and gram-negative. Many bacteria speak the AI2 language. And this was discovered by this lady, which is a very well-known scientist in the United States, because she does great science, but also because she is one of the greatest science communications I've ever seen. Her name is Bonnie Basler, and I advise you to watch her talk on TED.com on bacterial communication, because she is really a fascinating presenter. So Bonnie Basler was working on light production by Vibrio harveyi. It's a different Vibrio, which is a more complex network of quorum sensing. It's very complex. We're not going to talk about it. Just to give you an idea that it's very complex quorum sensing network. So Vibrio harveyi uses 3 different signals to produce light. 1, 2, 3. And all 3 signals come to the same response regulator to produce light. Okay. The fancy thing about the discovery of these is that the light of production by Vibrio harveyi can be induced by other bacteria. So this is a plate, and here you have Vibrio harveyi. It's a mutant that can only respond to the AI2 signal. And here, Bonnie Basler put E. coli and Salmonella, right? Salmonella and E. coli, they do not produce AHLs, so they were not expected to induce light production in Vibrio harveyi. But if you turn off the light, what happens? E. coli and Salmonella were inducing light production. They are talking to Vibrio harveyi, and they do that using the AI2 signal, okay, which is now known as the common language of most bacteria. Very important quorum sensing. Just very briefly, most of these signals go to the culture medium, but bacteria have also evolved ways to be sure that the signal will arrive to other bacteria. One of these ways is the production of membrane vesicles. These vesicles, you can see here, can be full of many different substances, but in the case of Pseudomonas, signals are sent to the culture media and to other bacteria using these vesicles. And nanotubes. So bacteria can connect with other bacteria and send molecules. Here you can see 2 mutants. This is mutant that is producing green fluorescent protein, and this one is a wild type. If you put the 2 cultures together, the protein starts diffusing from the cells, mutant cells, to the other cells. This is bacillus. So you can see that here, that how the green fluorescent protein is entering the wild type cells. And that protein is entering through these tubes, small nanotubes. So you may think, okay, but is this important at global scale, or just a few pathogens have evolved these mechanisms to attack us? So some marine microbiologists have made that question. How important is this? Only pathogenic bacteria use these mechanisms? Or maybe environmental, is it important in the environment? So they started to study this. Do any of you have an idea of what is this? Looks like galaxy, huh? Stars. Okay, closer look. This is with a microscope. Any idea? Very, very important habitat in the biosphere. No idea. This is called marine snow, or particulated organic carbon in the ocean. One of the most important organic carbon reservoirs in the ocean. These are small carbon particles that are sinking in the ocean. Okay? And there are a lot of bacteria that are growing in these small particles. You can see here the bacteria, and these small dots are the bacteria growing on that carbon. This is very important in the carbon cycle and in global heating phenomenon. Because we expect that when the CO2 increases in the atmosphere, then the CO2 fixation in the sea will increase, and we hope that this sinking mechanism will pump carbon from atmosphere to the bottom of our oceans. Okay? So these researchers, marine microbiologists, wonder, is it important quorum sensing in this habitat? And the answer was yes, very important. First they check if in these small carbon particles there were bacterial communication. And yes, there is a lot of AHLs, a lot of signal. So bacteria growing there are communicating. And then they check, what happens if we take these particles and we add the signal? The result was the enzymes, all the enzymes that are produced to degrade that carbon, they are increased when you add the signal. So that means that in these small particles, the bacteria are communicated to get coordinated and to produce the enzyme that degrade those carbon molecules. All right? So, we can say now that quorum sensing is an important phenomenon. Nobody believed it was, but now we have strong evidence that it's important in infection and it's important in nature. So the application of this is quite evident. If pathogens use communication to attack us, if we stop communication, we can stop pathogens. And that mechanism would be very different from antibiotic. When we use antibiotic, this is a very aggressive strategy. What we do is directly we kill the cell. And since it's very strong selective pressure, you will soon get resistance. And once you get resistance to the antibiotic, then you get again infection. Okay? But quorum sensing is different. Quorum sensing just deals with the expression of virulence, does not kill bacteria. Therefore, bacteria do not feel strong selective pressure if you act on communication. So, this is a virulent bacteria that is communicating and producing all that virulent factors. If you remove the signal, the bacteria will stop producing virulence factors. Okay? And it will be much easier for your immune systems to remove those bacteria. Okay? That strategy has been termed quorum sensing inhibition or quorum quenching. Stop bacterial communication to treat bacterial infection. Okay? And of course, nature has already done that job. So there are already organisms that can do that. This is an algae from Australia. It's called Delisea pulchra. And Delisea pulchra has developed quorum sensing inhibition strategy. the Lycia does not want the bacteria to grow attached to it. So what does she do? She produces a molecule that is very similar to the signal and blocks the receptor. What you see here is the algae, the seaweed, that has been located on a plate of a bacteria that is called chromobacterium biolacum. It's not important for you. This bacteria produces purple pigment only when it's communicating. So you can see here the algae is interfering with the communication, no signal, and therefore no purple pigment. Okay? In fact, the signal is there, just the bacteria receptors are blocked by the pyramid. Okay? So two mechanisms that we can use to inhibit bacterial communication. We can use molecules like the Lycia does that are very similar to the signal and block the receptor. It's called inhibition or antagonism. Or we can use enzymes that destroy the signal and prevent the receptor to receive the signal. Both strategies have been explored, and we have now proofs that these mechanisms are going to work. And I'm just going to show you a couple of examples that this works. So do you remember I showed you this slide? This is Pseudomonas aeruginosa, very important pathogen, human pathogen, produced biofilms. And this is a mutant, no quorum sensing. Infection capacity much, much lower. You can do this through quorum quenching. This is an experiment that we made in our laboratory with an enzyme, a new quorum quenching enzyme that we have isolated from a marine bacteria. This is again biofilm. So this is control. And this is not mutant. This is just standard culture where we put a quorum quenching enzyme. So you can see quorum quenching has avoided the production of the biofilm. Okay? That fish is called zebrafish, a very important animal model in biology. So these zebrafish were infected with a bacteria. The name is not important. That bacteria produced quorum sensing signals to kill the fish. Okay? So you can see if infected fish die, around 70% of the infected fish die. But if you give the fish a feed that contains a quorum quenching enzyme, the fish is protected from infection. And only around 20% of them will die. Okay? Last example. This is a plant. This is wild type. Oh, sorry. This is the opposite. It's changed. This is wild type. And this is transgenic. Okay? So, the plant has been infected with a pathogen, a bacterial pathogen. So you can see here the symptoms in the wild type. Okay? The plant is sick. But transgenic plant that is expressing a quorum quenching enzyme resists the infection because the bacteria cannot display the virulence factors. Okay? So, that is for chemical communication. We have great advances now in the biotechnological field to develop quorum quenching strategies for chemical communication. And we know a lot about it now. Some bacteria have extremely complicated chemical networks to communicate. Pseudomonas is one of them. We're going to see in a minute, Bacillus is extremely complex. E. coli is very complex, but we know a lot about it. Okay? And we are still already developing new drugs to use that strategy to fight bacteria. But physical is, we still do not know much about physical communication in bacteria. We know some phenomenon, and I'm going to show you 3 examples of physical communication in bacteria. They're very interesting, but first difference with chemical communication is that for physical communication, we do not know the molecular mechanism. For chemical communication, we know genes involved, receptors, molecules, everything. For physical, we know very, very, very little. So maybe we need a physicist to assist us with that, because it's probably too difficult task for biologists or molecular biologists. So 3 examples. The first example are biosonics, production of sound by bacteria. Many living organisms produce sound, and many bacteria produce sound. We don't know exactly what is the mechanism. Sound comes from vibration. And there are things in the cells, in bacterial cells, that indeed vibrate. Cytoskeleton proteins that are moving in the cells are vibrating. DNA that packed and unpacked is vibrating. Okay? But the intensity of that vibration is very difficult to produce a sound that we can measure. So probably there are several mechanisms that are acting together to produce a vibration that is high enough to be heard. Okay? This is a bacteria that produce and senses sounds, biosonics. This is Bacillus. This is very fascinating bacteria. This bacteria produces, have you ever heard of Bacillus? Do you know any Bacillus? Never heard about Bacillus? Bacillus is almost everywhere. If you do like this, in your hand, you have a lot of Bacillus. But some of Bacillus are very dangerous. Bacillus anthracis is involved in biological work. Have you ever heard biological work? Because the spores are extremely resistant and very effective. Okay, so anyway, Bacillus. What is particular about Bacillus? The production of spores, a resistant cell. So here you can see the Bacillus cells, and here the formation of the spores. That's a very, very complicated molecular mechanism. How to decide to make the spore, and how the spore will differentiate inside the mother cell. So here you have a Bacillus, and this is the spore that's being formed inside the mother cell. Quorum sensing is involved with that, and we know quite a lot on the mechanisms of formation of spores, because also there is population decision on that, and all the population decides we should form spores together. Okay? We don't know so much about germination. When you have a spore of Bacillus, how does it decide I should germinate? Well, the most surprising thing is that sound is involved in that. Okay? So this is Bacillus carboniferus. It doesn't produce sound that we can measure. Maybe some very, very loud sound that we cannot measure. But it can hear, it can listen. Right? So this experiment was, this is a Japanese University of Tokyo laboratory. Okay, a little bit, some years ago. So they put spores in a media that is not adequate for germination. And in that media, spores do not germinate. You can see the black dots, it's not germinating. But you can cheat bacteria. And how? Just with sound. So if you put that plate of spores in a media that is not adequate for germination, but you give them sound, the bacteria will understand that the media is correct and start germination. And not just any kind of sound. This is very specific, you see. This is the frequency that produces germination. 3 different sequences. Human air can fill up to here, more or less. This we cannot hear, but this is a sound that if it's strong enough, we could hear. Okay? Of course, this was very, very low intensity. Okay? So that was surprising. That the signal came from sound to germinate. And this was even more surprising. Who was producing that sound in nature? So Bacillus carboniferus is not producing the sound. At least it's one that we can detect with our microphones. But Bacillus subtilis produced sound. So if you use very special equipment to measure the sound that Bacillus subtilis is producing, this is what you get. Okay? This is control as a medium without Bacillus subtilis. And this is the sound that is being produced by Bacillus subtilis. The frequency. Okay? So you can see the maximum of the production of sound by Bacillus subtilis. Maximum for germination of Bacillus carboniferus is the same. The same. So the sound that is produced by Bacillus subtilis can be heard by Bacillus carboniferus. What is the hypothesis behind this? So, if Bacillus carboniferus is listening to decide to germinate, what he expects is that there are other bacteria close to it that are growing and producing the sound. So if the bacteria can hear the sound of other bacteria growing, then it can decide to germinate. So, bacterial communication. But, many questions. How do they feel? First, how do Bacillus subtilis produce this sound? Because it's very clear, the pattern is very clear. It's not specific. We don't know how is it produced. Very, very difficult that it's in a specific sound due to movement of cytoskeleton protein. And how does Bacillus carboniferus listen? No idea. We don't know. Okay. So this is one of the most physical, surprising physical phenomena. Okay. So this is biosonics. Now light. Light has a much smaller wavelength than sound. One of the surprising things about sound is that the wavelength of sound is much wider than a bacteria. So how can a bacteria can sense something that is wider than the vibration wavelength